I'm going to do a mini series here introducing the different steps of creating ATP and gaining energy in a cell. So we're going to look through a video on glycolysis right here, and then we're going to do oxidation of pyruvate, the TCA or the Krebs or the citric acid cycle. There's many different names for it. And then we're going to look at the electron transport chain and chemiosmosis. So first step in this would be glycolysis. So we will write that up here. This is the only step that occurs in the cytoplasm. So it is the only step that occurs in the cytoplasm, and it starts off with the 6-carbon monosaccharide glucose. So it starts off with just a glucose molecule, and then through 10 individual reactions that are each catalyzed by a different enzyme, it'll end with 2 3-carbon pyruvates. So we start off here with a 6-carbon glucose molecule. So if we look like there's 6 carbons in this, 1, 2, 3, 4, 5, 6, you will end, after 10 steps, you will end with 2 3-carbon pyruvate molecules, and you'll also get a few other things that I'll discuss in a second. But if we look, there is a few steps that are pretty important for this glycolysis that you should know. And if you need to know every step, then I encourage you to look at a textbook or a Google image that shows every single individual step and what goes on. But in this video, I'm just going to talk about the investment phase, as I like to call it, where you're investing energy into the reactions, and then the payoff phase where you're gaining energy back. So in step 1, we are going to write this in a different color here. So in step 1, when you start off with your 6-carbon glucose, you're going to invest ATP. So you're going to use an ATP in a breakdown for you. So you're going from glucose to what is called glucose 6-phosphate. And then nothing really happens until the 3rd step, where you invest another ATP. And that is when you're going from a fructose 6-phosphate to a fructose 1,6-biphosphate. And once again, I encourage you to look an image up in your textbook or the internet just to see kind of what I'm talking about here. But if you're looking at this at a basic level, all you need to know is that in steps 1 and 3, you invest in ATP. So you use energy in these steps. Then comes the step 6. So if we look at step 6 here, we'll do this in another color. We are gaining an NADH. So an NAD is being reduced, so it is gaining a hydrogen onto it. So we're going to put gain NADH, which will be important, as we'll see in the next coming videos. And then we're going to look at step 7 and step 10. So 7 and 10 gain ATP. So in each one of these steps, we're gaining 2 ATP. So we have a net gain of 2 ATP molecules since we used 2 here, and we're gaining 4 here from each one of these steps. So you gain 2 ATP from here and 2 ATP from here. So there we have your input of energy here at the beginning of glycolysis. So people often say you need to spend ATP in order to get ATP, and they're hydrolyzed in the first part. And then we have our payoff phase when we start to get the ATP back, and we end up with our net gain of 2 ATP. So the ATP that are made via substrate level phosphorylation, which happens in this step. So if I'm going to draw a little simple image here to help you out. So we have this enzyme here, let's say, that looks something like this. And we'll draw the second step here, same enzyme. And we have some sort of substrate right here, and that is attached to a phosphate group. And then we have our ADP right here. ADP, and this ends up going to here, where you get a product plus ATP. So you're adding a phosphate group. So this is adenosine diphosphate, and this is adenosine triphosphate. So there's an extra phosphate group here that is coming from right here. So this phosphate group is getting cleaved off and it is added here, and then this has no phosphate group attached to it. So this is your product, and this is your substrate. So this is substrate level phosphorylation. So the net gain, actually I do want to talk really quickly about this NADH. So NAD stands for nicotinamide, hold on, I'll write this down. Nicotinamide adenine dinucleotide. Nicotinamide adenine dinucleotide. And it's a non-protein cofactor. It's often referred to as just a cofactor. And it's an electron carrier. So it carries 2 electrons and 1 hydrogen. So when it's in its oxidized form, so meaning it has lost electrons, remember Leo the lion says Gur, so Leo stands for loss of electrons. Losing electrons is oxidation, and then Gur is gaining electrons is reduction, so reducing. So in its oxidized form, it has no electrons attached. So if we look here, oxidized, it would be NAD+. No electrons attached. And then its reduced form, it is NADH. So it has electrons attached. So our net gain from one glucose molecule entering glycolysis, I'll do this in red down here, our net gain is 2 pyruvate molecules. Pyruvate. 2 ATPs, 2 NADH, 2 hydrogens, and 2 H2Os. And I encourage you to write this down just in case if you need to remember it. So we're going in with a 1 glucose molecule, 2 NADHs are going in, and 2 phosphates are going in, and 2 ADPs. And then you end up with your 2 pyruvates, 2 ATP molecules, 2 NADHs, 2 hydrogen, and 2 waters. In the next video, I'm going to talk about the oxidation of these pyruvates before it enters the Krebs cycle.